Hello, everybody. Welcome back to Anatomy and Physiology. This week we are going to be uh, reviewing and moving along with the stress response. I want to clarify a few things that I said wrong in the previous lecture, and we will be talking about the control of blood glucose by insulin and glucagon. That's what we're going to do today. The next class will be discussing thyroid hormone and growth hormone in the context of somatic growth of your body, and metabolism of certain cells. And then we will do a very quick coverage of the endocrine control of water and salts, electrolytes like sodium, potassium, and calcium. So that's this week. Next week, we will turn our attention to the endocrinology of male and female reproduction, male reproductive endocrinology and female reproductive endocrinology. That's next week. So that's our roadmap for the lectures for for this week and next week. Before I go on, I have a few announcements. One is that I will grade problem set 3 soon. It's not graded yet. I haven't looked at them yet, but I will grade them as quickly as I can, and I will get them back to you graded much like I did for exam 2. The worksheets for lab is posted now. So they're available now, and they are due on Friday at 11:30 p.m. This worksheet you will see is a comprehensive review of brain anatomy. And so I want you to work on brain anatomy. You'll see the instructions on Moodle and on the document. And please let me know if you have any questions or if you have any issues with completing this lab worksheet that make you want to ask for an extension. Please just ask. Quiz 4 will post on Thursday, and it is due on Sunday. Let's see, I need to check those dates. Thursday is Thursday the 9th of April. That's when I will post it, and it will be due on Sunday the 12th of April at 11:30 p.m. Quiz 4 will cover endocrine names and effects, the names of cells, the names of hormones, the types of hormones, the effects of hormones, and quiz 4 will cover leg muscles that you studied on the worksheets for week 9 and 12. That's last week, and then the week before break. Those cover the leg muscles. And the coloring book is another source of information for leg muscle review, pages 60 through 64. So those are your sources for this week's quiz. I do want to make it clear that the lab worksheet is on brain anatomy, and quiz 4 will be on endocrine and leg anatomy. And so the brain anatomy is important to learn, especially for the final exam, but that's not what's going to be covered on quiz 4. Things have gotten a little out of sync, in part because of the strange world we're in now. I'll also comment that these narrated PowerPoints are now posted as video on the YouTube channel. So this, if you prefer to watch them as a video, which allows you to stop at any point, pause the video and take notes, as opposed to at the end or beginning of a slide, those are available to you on the YouTube channel, and that link is at the top of the Moodle page. Okay, so we're still talking about the stress response and the endpoint being cortisol and its effects. So let's go through this again because it's important, it's critical for life, and I made a mistake, so I want to clarify this. So stress, any stress, whether we're talking about a life-threatening stress of a bear hurtling at you, or chronic stress like worrying about whether to take grades or letter grades or credit, no credit grades. All of those are stressors and they have an effect on our body. So our brain detects stress. The brain processes stress and sends signals to the hypothalamus. The hypothalamus does many things, but one of them that we're talking about is there are neurons, neuroendocrine cells in the hypothalamus that project and release CRH at the median eminence. So they have axons from the hypothalamus proper into the median eminence, into the median eminence of the infundibulum, and there they release CRH into the portal circulation. CRH goes into the circulation, but specifically it goes from the infundibulum, the median eminence, into the anterior pituitary gland. It goes everywhere because blood goes everywhere, but this is the target of CRH, the anterior pituitary, specifically the corticotrophs of the anterior pituitary. Those cells are specialized to express the CRH receptor. Corticotrophs are stimulated by CRH to produce POMC and to metabolize POMC into ACTH. ACTH is released from those corticotrophs into the circulation. The blood goes everywhere. The cells of the adrenal cortex express the ACTH receptors. Stimulated by ACTH, they produce cortisol. Cortisol is released into the blood, goes everywhere, and its main effects are to increase blood glucose, which we will discuss in some detail today, to inhibit growth and reproduction, which we will not discuss today, and to inhibit immune responses, which is more immunology than it is anatomy and physiology. And in any case, I won't cover the molecular mechanisms of how cortisol is an immune suppressant, but it is a immune suppressant. Prednisone, a drug that is a cortisol analog, is often used as a drug to inhibit immune responses. So here is the flow chart for stress. This is another way of looking at it. As I said, stress causes the hypothalamus to release CRH at the median eminence. The CRH triggers the corticotrophs of the anterior pituitary to release ACTH. ACTH in the circulation goes everywhere, but it also goes especially to the adrenal cortex, where the cells, the cortical cells there, express the receptor for ACTH, and they release cortisol, which does the immune suppressant as well as causing an effect on metabolism, which I will discuss in a minute. The hypothalamus also has an effect on the posterior pituitary, and this is going to be important when we talk about vasopressin, but that's not until later this week. So keep that on hold. The stress, going back to the original stress, the stress also is detected by the sympathetic nervous system. The sympathetic nervous system releases norepinephrine and epinephrine, and it also triggers the adrenal medulla to release epinephrine. Epinephrine is released from the adrenal medulla as well as from the cells of the sympathetic nervous system proper. This stress response has many important things, including increase in heart rate and an increase in blood pressure that we talk about in AMP2. It increases respiratory rate that we talk about in AMP2. And now here is the point that I want to clarify. Cortisol and epinephrine both increase the effects well, they increase the effect of a hormone called glucagon, which is going to increase blood glucose and increase glucose production. Those hormones, cortisol and epinephrine, inhibit the effect of insulin. Insulin's main effect is to store glucose, store it as glycogen in the liver, store it as fat in fat cells, and store it as protein in muscle and other cells. So insulin's effect is to reduce blood glucose and store it for a later time. Glucagon's effect is to increase blood glucose and use it now. And so our stress response here and here is to increase blood glucose and increase glucose metabolism. So we release blood glucose from liver. The reason we do this is we're in a stress response. We evolved for an acute stress where our muscles need glucose in order to get us out of there. And our brain needs glucose in order to figure out how to get out of there, how to get out of that stressful situation. Our brain and our muscles need glucose in order for us to survive. And that's the target of these stress responses through the sympathetic nervous system and through the HPA, hypothalamus, pituitary, adrenal axis. If you have questions about this stress response, please post them to the forum or send an email to me, and I can post it to forum if I think it's of general interest. So let's do a fill in the blank. Ordinarily, if we were in class, I would show you this slide and I would stop and I would say you'd have a hard copy of this and you'd print it out. Well, you might not have a hard copy of this, but you can still write the answers. So what I want you to do is pause the PowerPoint. I'm going to stop it in a few seconds and write in this box, write the anatomical location of the hormone, of where the hormone is released from the hypothalamus. So here's the first step. The hypothalamus releases what hormone? Put that over here. And it releases it where? Put that in here. Then go on to talk about what happens in the pituitary, in the anterior pituitary gland. What is the name of the hormone? And what is the name of the hormone that is released by the anterior pituitary? And what is the name of the cell type and the receptor that triggers the release of this hormone? That hormone goes to the adrenal cortex, and I'm giving this to you. It's going to increase the production of cortisol. We will talk about feedback, negative feedback loops in a few minutes. And then here, write what you know about the effects of cortisol on liver, fat, and muscle, especially. And if you feel like it, write what it does to the immune system. So again, I'm going to stop this now. Take time to write these answers. Writing is an important part of learning, and so I want you to do this by writing down the answers, and we'll talk about it in a few minutes whenever you proceed with the PowerPoint. Here are my answers. The hypothalamus, the hypothalamic neuroendocrine cells release CRH in the median eminence, and it's released of the infundibulum, and it's released into the portal circulation in the median eminence. Blood flow carries that CRH to the anterior pituitary gland. The anterior pituitary corticotrophs express the CRH receptor, and when stimulated by CRH, they produce ACTH from POMC, from pro-opiomelanocortin. ACTH is released from those corticotrophs into the systemic circulation, blood flow. Adrenal cortex releases cortisol. Cortisol goes into the blood. At the liver, it triggers the release of glucose. Well, here's some details about that. The liver cells perform gluconeogenesis, that is to say, they synthesize glucose from other carbon sources, and they perform glycogenolysis. They hydrolyze the starch, the form of glucose that we store as starch, glycogen. This process of glycogenolysis is stimulated by cortisol, and so glycogen is broken down into its component parts, glucose, and this glucose is, so the glucose is either synthesized de novo or is produced from the hydrolysis of glycogen and released into the blood so that the muscles and brain can work at maximum efficiency. Fat cells are stimulated to metabolize fat, take it out of storage, and produce glucose. Muscle cells are told, okay, don't store glucose as glycogen, and in fact, maybe, depending on the timing, hydrolyze protein into glucose. We need protein in order to do an efficient muscle contraction. So this is not purely productive, but it is an effect of long-term stress. And as I said before, the immune cells are suppressed, and we're not going to talk about the details of that effect, but it is there. So that's the slide to describe the feed-forward signaling that and the effect of cortisol. Now I want to remind you about POMC, pro-opiomelanocortin. We talked about this in the context of peptide hormone processing, but now we need to be specific. So here is a map of POMC. Here's the N-terminus of POMC. Here's the carboxy terminus of POMC. It's a protein. It's got 260 amino acids. POMC is the protein that is synthesized by the corticotrophs in the anterior pituitary. The stimulation of the corticotrophs by CRH leads to an increased production of POMC and an increased processing of POMC. So POMC, when it is cleaved, pro-opiomelanocortin, when it is cleaved here and here, it produces ACTH. And there is the hormone that we've been talking about for the control of cortisol. When it is cleaved here and here, it produces gamma MSH, melanocyte stimulating hormone. And here it produces alpha melanocyte stimulating hormone. And here, beta melanocyte stimulating hormone. So alternative cutting of pro-opiomelanocortin produces the various forms, alpha, beta, and gamma, of melanocyte stimulating hormones. Now you may know how to unpack the word melanocyte. Melanocytes are skin cells that produce pigmentation. Skin pigmentation is due to the production of melanin in the melanocytes of the skin. In addition to producing ACTH and MSHs, alternative cutting of pro-opiomelanocortin produces the enkephalins and endorphins. Here is beta endorphin, and here is enkephalin. So some of the opioid proteins in our body relating to pain are produced as alternative cleavage products from pro-opiomelanocortin. So what I want you to do now is stop the narration and write what I just told you. Write in here the major products of pro-opiomelanocortin cleavage. I'm going to stop so that you can take a moment to write out their names. And here are my answers. So Pro-opiomelanocortin is a pituitary prohormone produced in the corticotrophs of the anterior pituitary. We have alternative cleavage of POMC. And so what does it produce? It produces ACTH, which is the abbreviation for adrenocorticotropic hormone. It produces various forms of melanocyte-stimulating hormone, and it produces the opioid peptides beta-endorphin and met-enkephalin. So those are things to know about POMC cleavage. So let's apply some of your knowledge now of the cortisol signaling in the context of a clinical case. So Addison's disease, which is also known as primary hypocortisolism or adrenal insufficiency. This is when the adrenal gland doesn't produce enough cortisol, hypocortisolism. And it's primary because we're talking about a defect in the adrenal gland. So what are some of the symptoms of low blood cortisol? Fatigue and weakness, weight loss, and here's an interesting one, hyperpigmentation of skin. I'm going to ask you to think about that in just a minute. So we will talk about why do we get skin pigmentation with adrenal insufficiency. So what are some of the causes of adrenal insufficiency? Well, one can be an autoimmune destruction of the adrenal cortex. If our immune system has somehow been inappropriately programmed to destroy the cells of the adrenal cortex gets messed up and it doesn't produce enough cortisol. Some infections can lead to immune destruction of the cortisol of the adrenal cortex, or also just the bacteria can destroy the adrenal cortex. And there are genetic forms of primary hypocortisolism, genetic defects that cause the adrenal glands, the cells of the adrenal cortex, to produce insufficient cortisol. Enzyme defects in the enzymatic pathways that lead to cortisol production. There is a phenomenon or a syndrome also known as secondary hypocortisolism. So hypo, low blood cortisol, hypocortisolism, but it's secondary if the defect is not in the adrenal gland, but if the defect is in the corticotrophs of the anterior pituitary. That would be secondary hypocortisolism. If the corticotrophs are not producing enough ACTH, whether usually for genetic or mostly genetic reasons, if the corticotrophs are not producing enough ACTH, that would lead to secondary hypocortisolism because the adrenal would not get enough signal to produce cortisol. So now I want to go back to the hyperpigmentation of skin. Why would hypocortisolism produce too much skin pigmentation? I want you to think about this, write down your answer, and then pause the narration, write down your answer, and then move on. The answer to hyperpigmentation of skin is this. It has to do with lack of negative feedback. So if there is low cortisol, there is a lack of negative feedback. Low negative feedback. So if there is low negative feedback, then there is less negative feedback of CRH production, so there is an increase in CRH production. CRH stimulates the corticotrophs of the anterior pituitary to produce ACTH. So it produced increased stimulation of corticotrophs to make ACTH. But remember, ACTH comes from pro-opiomelanocortin. So the stimulation of the anterior pituitary gland to make ACTH also increases the production of MSH, alpha, beta, and gamma melanocyte stimulating hormone 2, because they are produced from the same precursor, POMC, as ACTH. So increased CRH and increased stimulation of the corticotrophs to make ACTH has a secondary effect of increased production of MSH. Increased production of MSH leads to increased proliferation of melanocytes and their production of melanin. And so in primary hypocortisolism, you get an increase in skin pigmentation, especially in areas like the folds of the neck and the inside of the elbow. Areas that don't usually get pigmented are often pigmented in the case of primary hypocortisolism. Now, you might get this in secondary hypocortisolism too, where, no, that's not true. So this is just an effect of primary hypocortisolism. Let's talk about the opposite of hypocortisolism, which is hypercortisolism, too much cortisol. And this syndrome has a name called Cushing's syndrome, after some doctor who described it for the first time. The causes of hypercortisolism can be a tumor of the adrenal gland, so there's adrenal cortex enlargement and too many cells of the adrenal cortex. So a tumor or an adenoma of the adrenal gland can cause too much adrenal gland cells, and that would lead to too much cortisol. It can also be caused by a adenoma, a tumor of the pituitary gland, where the tumor causes the proliferation of corticotrophs. So there's too much corticotroph, so there's too much ACTH production, so there's too much stimulation of the adrenal to make cortisol. So both of these, or either of these, can cause hypercortisolism or Cushing's syndrome. And the symptoms are complicated, and we're not really going to have time to go into the mechanisms. They cause a very particular form of obesity, which leads to fat deposits in the upper body, chest and abdomen. The fat can deposit in the face, and it causes a syndrome or a sign called moon face. The face gets big and round. Hypercortisolism can cause slow growth, and it can cause various other symptoms that are hard to explain and would require a lot more time for me to try to understand and explain them to you. So now let's move on from the stress responses to metabolism, specifically of control of blood glucose. And then we will not do growth and metabolism today. We'll do growth and metabolism and water and electrolyte control in the next class. So now we're going to turn our attention to metabolism and the control of blood glucose by insulin and glucagon, remembering that cortisol inhibits the effect of insulin and magnifies the effect of glucagon on blood glucose. So let's talk about insulin. Insulin is a peptide hormone. It's made from a pre-proinsulin. So pre-proinsulin has the signal sequence. When the signal sequence is cut off, the pre part is cut off. So after cutting of the signal sequence, we now have proinsulin, and then that proinsulin is cut to make insulin. So here is pre-proinsulin. Here is the amino terminus of pre-proinsulin. Here is the carboxy terminus of the pre-proinsulin. This is just showing the generic structure of this peptide protein. When the signal is cut off, there's the lack of the signal here. Now we have proinsulin. And when these linking parts are cut, we now have insulin. And insulin is formed from two chains, the A chain and the B chain, that both come from pre-pro insulin or proinsulin, and they are held together by disulfide bonds. So this is the structure of the insulin molecule that is formed from two chains, the A and the B chain, both of which came from the proinsulin, and these two chains are held together by disulfide bonds. The stimulus for glucose, the stimulus for insulin, is high blood glucose. So when you eat a meal and your digestive tract digests the nutrients that you ate, glucose goes up. This is the subject of a section in AMP2. Glucose increases in the bloodstream, and that is detected by the cells in the pancreas called the beta cells. So the beta cells of the pancreas detect high blood glucose and are stimulated by that. They are stimulated to release insulin. The insulin has effects on liver, fat, and muscle. The insulin interacts with a tyrosine kinase receptor, an RTK, and the activation of the tyrosine kinase receptor by insulin causes glucose uptake, and glycogenesis, the synthesis of glycogen to store that glucose, to uptake and store amino acids as protein, and to synthesize glucose and protein really into fats in fat cells. So after a meal, our body takes the blood glucose and stores it as glycogen in fat and muscle, no, in liver and muscle, and stores it as fat in fat cells. Here is a slide that describes the mechanism, the cellular and molecular mechanism for how glucose stimulates the release of insulin by pancreatic beta cells. So this slide summarizes a lot of the basic concepts of anatomy and physiology, goes all the way back to cell physiology. And I like this because it's taking a topic fairly late in ANP, and at least ANP1, and circling back not just to the beginning of this course, but circling back to cell physiology. So I've blanked out here certain parts of this slide that we're going to walk through. So the first thing I want you to do is fill in this circle here. Well, let me give you a hint. So here, this is a cartoon of a pancreatic beta cell. So that's what this is supposed to be. This pancreatic beta cell has transporters expressed in the surface membrane. These are glucose transporters. They are uniporters. They do facilitated diffusion of glucose from the blood and the extracellular fluid into the cell. So we're going all the way back to cell phys, talking about facilitated diffusion of glucose through a specific uniporter. the GLUT2 glucose transporter. I do not care if you memorize the name of this protein, but I do want you to know that facilitated diffusion is how elevated blood glucose after a meal gets into pancreatic beta cells. So now we have an increase in glucose going into the cells. The next step here is ATP. So I want you to pause the narration and write what goes on in this circle that leads from glucose to the production of ATP. So I have revealed here what was under my circle. Hopefully you have more detail than this slide provided. So the glucose is metabolized by glycolysis. So glucokinase, I think we called it hexokinase in cell physiology, produces glucose 6-phosphate. And remember, there's many steps of glycolysis, some of which you might remember. The glycolysis produces, let's see if we can hit the big points here, and I'm doing this from memory too. Glycolysis of glucose produces pyruvate. Pyruvate then is oxidized into, well, CO2, NADH, and acetyl-CoA. Acetyl-CoA then becomes the substrate for the Krebs cycle. The Krebs cycle produces a little bit of ATP, really GTP, but that can be easily transformed to ATP, but it produces a lot of NADH and FADH2. Those high energy electrons of NADH and FADH2 are fed to the electron transport chain of oxidative phosphorylation, oxidative cellular respiration. The electron transport chain converts the energy of high energy electrons in NADH and FADH2 into a proton gradient across the mitochondrial inner membrane. Then that proton gradient is transformed by ATP synthase into ATP. ATP, right there. So there's a lot of cell physiology that is embedded and only partially revealed in this part of the slide. I will not test you on cell physiology, on metabolism, on glycolysis, on Krebs cycle, on electron transport chain, on ATP synthase. You've already been tested on those, but here they are again. So ATP goes up because the glucose concentration in the cytoplasm went up. The ratio of ATP to ADP goes up. ATP is produced, ADP stays pretty much the same, so this ratio goes up. So now let's move on to the next step. The level of ATP has increased, and I need to introduce the next player, the ATP blocked, which is also usually called the ATP sensitive potassium channel. There are potassium channels, so this is a new type that I'm introducing. It is a potassium selective channel that is blocked by ATP. So now I want you to think about and fill in what happens in this blob. What happens to the cell's membrane potential when the ATP blocked potassium channel is blocked by ATP? Think about what happens to the membrane potential of the pancreatic beta cell when this channel is blocked by ATP. Stop. You're going to think and write the answer and then resume. Here is the reveal. The block of the potassium, of the ATP blocked potassium channel, causes a decrease in potassium efflux. So we have a decrease in potassium efflux from the cells, and a decreased potassium efflux leads to depolarization. Depolarization of the membrane potential is what follows the block of this potassium channel. That should be clear to you. All right, so now let's go on to the next step. What does this depolarization do that is going to cause the exocytosis of granules that store insulin? So the yellow circles are supposed to be insulin, and that is released by this process that we're about to reveal here. I hope you know what the green circles are supposed to symbolize. Depolarization leading to exocytosis. What goes in this circle? Stop it, write it down, and then move on. Here is the reveal that I really hope you got pretty intuitively. Depolarization causes the activation of voltage-gated calcium channels. The green circles are supposed to represent the influx of calcium. Calcium binding to synaptotagmin, conformational change of snares, V snares and T snares, cause the calcium-dependent exocytosis of insulin into the bloodstream from the pancreatic beta cells. So you should probably go back and review the story of how increased glucose concentration and increased glucose uptake causes this series of effects that leads to insulin release. Summarize it for yourself. I'm not going to spend time to repeat it, but I think you should. Now, insulin released into the blood has effects. It has main effects on muscle and liver cells. So let's talk about the cellular and molecular mechanisms for what insulin does to muscle and liver cells. Well, so the beginning point is insulin binding to insulin receptors, and the end effect is glucose uptake by the by the liver and muscle cells, fat cells too, and increased glucose utilization. That is to say, synthesize glycogen. This is storage of glucose. Increased glycogen synthesis, increased lipid or fat synthesis, increased protein synthesis by the cells of the muscle, liver, and fat. That's the end point. Insulin is the beginning point. And what I want you to do is fill in the missing steps that are here. So what this slide shows is that activation of the insulin receptor, which is cartooned here, causes a generic signal transduction, but I want you to unpack what that is. That causes gene expression and regulation. So increased expression of a glucose uniporter and increased expression of the enzymes necessary for glucose storage as glycogen, lipid, and or protein. But there are things that are missing here. So I want you to fill in this box here with what is not shown on this slide. There are things that you know that whoever drew this cartoon didn't include that you should include now. Stop, write it down, and then proceed. So here is what I wrote down for these missing steps. Let me narrate this a little bit. Insulin binds to the insulin receptors. That binding of insulin to the insulin receptors causes dimerization. I didn't even include that in my own answers. Dimerization of the insulin receptor subunits. The dimered receptor subunits phosphorylate each other on tyrosine residues, tyrosine side chains. cross-phosphorylation. This phosphorylated subunits, these phosphorylated subunits recruit scaffolding or adapter proteins that recruit and activate a small G protein called RAS. RAS activates MAP kinase, which activates a kinase cascade. This cascade of kinases, all of which are tyrosine kinases, that is to say they phosphorylate proteins on tyrosine residues, leads to signal transduction, and some of them are transcription factors, so they change gene expression. So these are most, but not all, of the missing steps of this cartoon. Dimerization, cross-phosphorylation, adapter protein recruitment, RAS activation, MAP kinase activation, and the activation of a kinase cascade. This leads to many things, including the production of another glucose uniporter that helps bring glucose into the cells, and it increases the enzymes that cause the storage of glucose as glycogen, fat, or protein. Again, I don't care for you to memorize the name of this uniporter, but it is a facilitated diffusion transporter that brings glucose. Remember, glucose is high now because we've just had a meal that brings the high concentration of glucose into the liver, fat, and muscle cells. On the flip side is glucagon. Flip side of insulin is glucagon. So let's go through this in a little less detail than we went through with insulin. Glucagon is a peptide hormone. From pre-glucagon, there is no pro-glucagon. So when the signal sequence of pre-glucagon is cut off, it produces glucagon. Doesn't need any additional cutting. The stimulus for glucagon release from the alpha cells of the pancreas. So the organ that releases glucagon is the pancreas, but the cells are different. It's the alpha cells that produce glucagon and release it, whereas it was the beta cells that produce insulin and release it. The stimulus of those cells is low blood glucose, and we're not going to go into how that is detected. The target cells are the same as the target cells for insulin. The receptor for glucose is a GS-coupled GPCR, and through activation of GS and its downstream effects in liver, fat, and muscle cells, there is a breakdown of glycogen, glycogenolysis. There is a breakdown of fat. There is, and both of those are going to cause an increase. Both of these things would cause an increase in the production of glucose from glycogen or from fat. The uptake of glucose by liver and muscle is decreased so that that glucose can be in the blood for the brain. The muscle is producing its own glucose from glycogen. And there is decreased amino acids uptake and synthesis because during this response, if we're in low blood sugar conditions, we don't want to be synthesizing protein. We want to be making sure that we keep the blood glucose high enough for our brain to function. So that's how glucagon works. And that's the information I want you to know about glucagon. And before I end, I want to use what we've just learned about insulin and glucagon to talk about homeostasis. So let's do this. This is somewhat conceptual. Let's do this systematically. So insulin and glucagon are antagonistic. They work against each other. Glucagon works to increase blood sugar, say when you're fasting, and insulin works to store blood glucose and decrease it, say after a meal. Those two effects are against each other, and so they're called antagonistic systems. So let's see what happens. I like this conceptual way of thinking about antagonistic control systems. So let's think about it. The way I think about this is I put the controlled variable, the thing that is controlled, on the x-axis, and the thing that has power to control that variable on the y-axis. And there is going to be a set point. So let me use a simple example that becomes a little less conceptual and maybe a little more intuitive. Let's think about how your house, your room, the classroom, if we were sitting in the classroom, how that room controls the temperature. So the controlled variable is the indoor temperature. It has a set point. There's the thermostat sets the inside temperature at some set point. Let's just say a moderate temperature of 70 degrees Fahrenheit. The room has a heater and it has an air conditioner that are antagonistic. The heater works to raise the room temperature and the air conditioner works to lower the room temperature. So the power to the heater or to the air conditioner is what is controlled by the thermostat. So let's see what happens. If the indoor temperature gets cold, if the indoor temperature were to get cold, the power to the heater would be high and the power to the air conditioner would be low. High power to the heater and low power to the air conditioner would cause the indoor temperature to rise. And so we would rise back towards the set point. If the temperature got too high, if it got much higher than the set point that controlled by the thermostat, the power to the air conditioner would be high and the power to the heater would be low. High power to the air conditioner, low power to the heater would cause the temperature to decrease towards the set point. At the set point, the power to the two antagonistic control systems is equivalent. And so the heater and the air conditioner are receiving equivalent power when we are at the preferred set point temperature. Deviation of the set point temperature above, deviation of the temperature above the set point triggers an effect that brings the temperature back down. Temperature, if you were to say open the window and cool off the room, say open the window in the wintertime, and that would cause the room to cool off, that would increase the power to the heater and decrease the power to the air conditioner, and the temperature would then, the system would try to raise the temperature back to the set point. And the set point is where they are of equal power. So now let's go back to physiology and think about insulin and glucagon. We have a set point for serum glucose. Our body wants to have blood glucose at a set point of about 100 milligrams per deciliter, which is the same as 1 milligram per mil. That is a common normal blood glucose. The power here is not a power, it's the hormone concentration. If the serum glucose were to gets high, let's go with serum glucose gets high, say after a meal. That stimulates insulin production and inhibits glucagon production. The stimulation of insulin production and the inhibition of glucagon production causes the causes the system to work back down. So serum glucose would be decreased because the increased insulin and the decreased glucagon would cause the liver, fat, and muscle cells to increase glycogen synthesis, to increase fat synthesis, to increase protein synthesis, to remove the glucose from the blood and to synthesize it into glycogen, fat, and protein. And that would lower the blood glucose towards the set point. If the blood glucose were to get too low, say during a fast, no eating, then the body is going to try to raise blood glucose because your brain needs glucose even if you're starving. So to do that, low blood glucose increases the release of glucagon from the alpha cells and decreases the release of insulin from the beta cells. Increased glucagon and decreased insulin causes increased glycogenolysis, increased lipolysis, and decreased protein synthesis. And so those things, the breakdown of glycogen, the breakdown of fat, is going to tend to increase blood glucose back to its set point. So glucagon and insulin are the antagonistic controls that help keep us at within a fairly a fairly narrow range of blood glucose concentration around the set point. There are, of course, diseases of this. Diabetes is a chronically high blood glucose that can either be due to a defect in the set point or is commonly caused by a defect in insulin production, that's diabetes type 1, or a decrease in insulin response, that's type 2 diabetes. Okay, so that covers metabolism, especially like glucagon and insulin. Oh, so I just made almost got confused. Remember that glucagon, let me just teach one more thing before we're done. Glucagon is the hormone. Glycogen is the glucose storage molecule. Do not get glucagon, the hormone, the peptide hormone released by the pancreatic alpha cells. Do not get glucagon confused with glycogen. Glycogen is the storage molecule for glucose. It is a starch. It is a what do we call that? An oligo, it's a carbohydrate. It is a polymer of glucose. Glucagon is the hormone, glycogen is the storage molecule. We'll pick this up again on the next PowerPoint.